Okay, in this case study, what I would like to do is to give you kind of an introduction to molecular modeling and how we can determine the stabilities of different kinds of molecules that you've learned about in your lecture class to this point, using some different types of computer calculations and this whole area of visualization along with doing the calculations is really called molecular modeling or computational chemistry. So first of all, molecular modeling then is to use different computer quantum mechanical calculations to perform what are called structure energy calculations and then visualize the results. And you've seen pictures like this in your general chemistry, your organic chemistry, even some of your biology, genetics textbooks that I'll show you a little bit later on. So molecular modeling and computer calculations, what they do is they allow organic and bioorganic and biochemists and molecular biologists to visualize molecular structures, predict the properties of molecules without going into the lab. So we can get a general idea what their shape, what their energies might be like. This is extremely important in the area of drug design. It's also a very important area in terms of trying to figure out drugs that might be able to interact with active sites of enzymes or DNA or RNA, which I'll show you a little bit later on with the coronavirus, which is why this is not live and basically an online module. We can also look at molecules that haven't been synthesized and may never be synthesized. What would their physical properties, what would their structures and energies be? So you've seen pictures like this in your organic textbook, in your general chemistry textbooks. You've seen different molecules in what are called the space-filling models, where you're actually looking at the van der Waals radius of the atoms. In real life, the molecule would not be color-coded, so this would just be a big blob, but it would have a generalized shape. We can also do in organic chemistry, you've seen these kinds of pictures where you have charge density maps. So for instance, this is going to be the structure of a tertiary carbocation. Where's the positive charge? It's over the center carbon, it's in blue. And then you can kind of see the hydrogens over here have a slightly different color. So in general, blue is partially positive in this program and red would be negative. So we can see those charge density maps, which we really can't see using any other method, and they can help us to evaluate some of the where the carbocations are, or actually what the most stable resonance structure of a molecule would be. We can also do visualizations to look where the lone pairs are. This is an oxygen. It kind of has a it has sort of a, I don't know, it's a slice of an orange type of a shape. This fluorine is at the end of this 2-fluoroethanol molecule. All three of the lone pairs on the fluorine make like a mushroom cloud. And so we can look at where the lone pairs are and looking at, look at where the p orbitals, or in this case, the sp3 hybrid orbitals for the fluorine and the oxygen would be. Also with these calculations, we can predict NMR spectra, we can predict IR spectra. So we could put a molecule in and we could get a prediction of where we would see the peaks in the IR or even the NMR. These calculations are getting better and better all the time, so that while they're not a substitute for the real physical values, they're getting closer and closer to be able to predict the true chemical shifts and true frequencies in IR. So there's a lot of different calculation methods that you can do. We're not going to talk about the quantum mechanics, the Schroedinger equation that you may have learned about a little bit in general chemistry. You would learn a lot more about it in physical chemistry or in a quantum chemistry class. We're going to treat these, sadly, we're going to treat them sort of like a black box, but they will give us results that we can go ahead and use to interpret structures or look at structures and ask questions and get the results that will help us solve those kinds of problems. And each calculation has its advantages and disadvantages. I'm not going to spend a lot of time on that, but I am going to give you some general ideas about which ones could be used in which circumstance. And that allows you then to use the right calculation to solve a particular problem. Now, I first saw computational chemistry on what was at the time $1 million computer in the basement of the Roman Haas Company, which I don't know if that exists, but they were bought out by somebody in Philadelphia on a senior class trip in, let's just say, the mid-'80s. And in the basement of this research facility, they had this one computer, and they talked about how you could draw structures and how they would be able to design. They were designing biocides and herbicides, which were used for plant as materials to keep plants from developing diseases. And that was the first time that I saw it. In graduate school, I did a little bit of this work. I never would have imagined that I could sit here at my table and have a computer that would kick the butt out of even that $1 million computer now. And there's even faster ones. Dr. Bruce, who this is her area of research, she has a computer that I think is almost as fast as the supercomputers that existed back when I was in graduate school. So you can do this you can do it on an iPad at a low level. But let's talk a little bit about the simplest calculation, which is still used and still pretty effective, and that's what's called molecular mechanics. Every technique has its own acronyms. Molecular mechanics includes things like molecular MM3, molecular method 3, Sybil, Amber. Those are programs that, for instance, Dr. Bruce's research group uses to look at very large molecules like proteins and enzymes. And the reason that you use these for large molecules is they are fairly simple, they're fast on a computer, you can do the most number of atoms, and you can do biomolecules, like I said, DNA, RNA, enzymes. You could also do polymers. But the problem is that it's a totally parameterized method. And what I mean by that is that the parameters that the calculations use to figure out the energy and the structure have to be taken from experimental results. So there have to be parameters for the molecules in order to develop these computational methods. And therefore, you're not able to do carbocations, you're not able to do metals, and you're not able to do molecules that haven't been synthesized and don't exist, because there's no parameters for those. So basically what happens is there are parameters for bond distances, bond angles, and steric repulsions. And those parameters will relate to the bond distance or angle, and they relate those values to energy. So for instance, here's a real simple calculation, or part of the calculation. So the bond energy for a carbon-carbon bond, let's say for a carbon-carbon single bond, it's somewhere in the neighborhood of about, and give me 0.5 angstroms either way. I think it would probably be about 1.6-ish angstroms. And so if you know what the optimum carbon-carbon bond length would be, what you can do is, by looking at a number of different molecules, you can determine a sort of a force factor that says, you know what, if I change that bond angle or that bond distance so many angstroms. If I stretch it out or if I try and compress it, it's going to cost me that much extra energy, either to stretch the bond or to compress it. And so if you take that and you calculate the bond energy for all the different molecules, you can then change the structure and recalculate the energy and see, did the molecule get more or less stable. So bond energy is one component. You can also have the bond energies, the angular energies. So in other words, if I have a carbon-hydrogen bond in a tetrahedron, I definitely know what that angle is. Ideally, that's 109.5. But if I'm going to distort that by either spreading it or contracting it, it's going to cost me so much energy per degree. Torsional angles are basically what we have in the sawhorse representation. So in the sawhorse representation, basically knowing that these groups right here are 180, if these were large groups 180 degrees away from each other, then they would have a certain most stable energy, and then as I bring things closer, well, as I start to stretch out that torsion angle, those torsion angles, it's going to cost me energy. And then the idea of steric repulsions, if you have methyl groups that are basically gouged to each other, that's going to be so much energy to have those two groups sterically close to each other, whereas if I start to move that methyl group away, I'm going to end up with less energy. So what we do is we make a geometry of the molecule and we sum up all those energies. Then the computer will change the structure a little bit and recalculate the energy. And the idea is that as we change the structure, the total energy of the molecule begins to go down. And at some point we find a geometry that is what we call the minimum energy. Now the problem is that if you start the molecule out here at a peak, this isn't a two-dimensional space. It's more like an n-dimensional space. So you have to search out in all directions, change the geometries a lot, and then try and figure out what the minimum energy is. And typically what we'll do with a molecule, if I'm trying to find the most stable conformation, is I'll just randomly generate 100 random structures, minimize each one so that hopefully I find what is called the global minimum, in other words, the absolute lowest energy. But it's not even a three-dimensional problem, it's an n-dimensional problem. So that is a little bit complicated, but the beauty is that the computer programs now will pretty much do that, and you don't really need to know that much, although you can get yourself in huge trouble if you don't quite know what you're calculating. So once the total energy is calculated, then the computer moves the position of the atoms, recalculates the energy, and then eventually we find a total minimum total energy. And then we can look at the structure that was predicted or that was calculated. So in terms of molecular mechanics, Sybil, Amber, they're very fast. They don't require expensive computers. You can use them on huge molecules like bioorganic molecules and polymers. The problem is you can't use them for molecules where there are no parameters, including carbocations, carbanions, metallic species, molecules that just haven't been synthesized, because this is all parameters based on experimental results. It also doesn't give you any information about orbitals or atomic charges, and so I still use this in order to basically screen conformations. Our next highest level up is what's called semi-empirical. And there are different versions of this. We'll do some calculations with PM3. there's AM1, I believe there's a PM5 now, there's MNDO. There's a bunch of these different parameters, but they all fit into what are called semi-empirical calculations. Semi-empirical calculations is a method that's hybrid. It has both parameterization and it also uses a bit of the Schroedinger equation. And so it's the same basic principle. Only now what we have is now we're kind of getting our energies and our parameters from partially using a Schroedinger equation, which means that I don't need any experimental results, but also it does need parameterization because to use purely the Schroedinger equation would be a lot more computationally expensive. It does require more computational time, and so smaller molecules have to be used. And since the method's based on wave functions, which is what the Schroedinger equation describes, you don't have to identify bonds. So we can do carbocations, we can do carbanions, we can even do radicals if we'd like. So we'll do an example of that where we'll calculate what the energy difference is between primary, secondary, tertiary, and an allylic carbocation. So I can do that at the semi-empirical level. The results can be charge distribution. You can end up with electron density maps, which I'll show you. In quantum chemistry, you would talk about HOMO, which is the highest occupied molecular orbital versus the lowest unoccupied molecular orbital. You may have talked about that in general chemistry or seen it in your organic textbook. And we can use semi-empirical calculations to predict the regiochemistry of the reaction. Now, since some of the parameters are used, then if there's no parameters for that molecule, then the results may not be accurate. For instance, metals are not necessarily handled well by the semi-empirical calculations, although my research area of interest in graduate school and as a postdoc was lithium, and it actually has very good parameters for lithium, but if I want to go to sodium or potassium, I'm out of luck because those metals haven't been parameterized very well. Although I will show you the program that I'm using does have some decent parameters for some of the metals. So then if you want to go, okay, I don't want any parameters, I want total Hartree or total Schroedinger equation, then you go what is called ab initio. Ab initio methods are things like what are called Hartree-Fock. DFT stands for density functional theory, which was just beginning to be developed when I was in graduate school in the late 80s. Nowadays, I can do it on this computer. It will take probably 2 or 3 hours to do a small molecule, but it can be done. Back in the day, there was no way to do this outside of a supercomputer. It's the most complicated of the calculations. It requires the most computational time. There was a time when you could only really do these effectively on a supercomputer. And, you don't have to have a supercomputer in your office. What you would do is link to a supercomputer within your region, like Ohio State has, I believe, a supercomputer that you can upload jobs to, and it would then do those calculations and send you back the results. This is totally based on Schrodinger's equation and wave functions. In general, it gives the most accurate results. You have to be careful that when you're doing these calculations, you have to make sure that the results are reasonable, which means you probably, if you're not an expert in this area, you should probably get help from an expert. And structures sometimes can just blow up, meaning they can be just totally unrealistic. So that's the highest level, and I can actually do that for small molecules on the computer system I'm using, which is only an it's an i5 built for virtual reality. So I mean, I can do these relatively quickly, and you'll be surprised how quick the calculations are when we get to do those. All right, so normally what we would do is we would be in the Carabine lab, and I would say, okay, everybody, let's go ahead and draw a methanol. well, I'm not in the Carabine lab, I'm at home, so let's